Hello, I am Stefano Sonedda and I am a PhD student in the Electronic and Computer Engineering program. In this presentation, I will talk about integrated electronics for molecular biosensing. Bioelectronics is the application of electronic engineering principles to subjects like biology, chemistry, or medicine. The union between such different subjects is made possible by biosensors, which are devices that can convert a biological response into an electric signal. During my PhD activity, I focused on different approaches to sense biological reactions by using integrated electronic platforms. The first part of my research focused on telomerase activity detection. Telomerase is an enzyme which causes the elongation of telomeres that constitute the terminal part of chromosome. Telomeres protect the chromosome during DNA replication and get progressively shorter. In human somatic cells, telomerase activity is negligible. When telomeres become shorter than a critical limit, the cells stop replicating and start its senescence phase. However, a strong telomerase activity has been found in cancerous cells, which are characterized by an uncontrollable and infinite replication. For this reason, telomerase can be used as a marker to detect the presence of cancerous cells and tumors. To detect telomerase activity, we developed a lab-on-chip CMOS device which contains all the tools needed for the measurements. In fact, telomerase detection requires three different steps. The first one is the functionalization of the sensing area with DNA probes. Then, we need to add the solution that contains the telomerase on the device and wait until the reaction happens at a specific temperature. Finally, we can electronically measure the changes in the telomere's length. The functionalization is obtained by an electrochemical procedure which binds DNA probes on the sensing area by using a potentiostat. Since the temperature is also a critical factor, a temperature control system has been implemented by using a heater and a temperature sensor. Finally, we have the sensor, which is implemented to a charge-modulated FET, a device that can sense charge variation on its sensing area. The CMFET is a floating gate MOSFET whose gate has been partially exposed. This exposed part uses sensing area. Charge variation on the sensing area can cause a shift of the device threshold voltage, which in turn changes the device current. The presence of telomerase brings positive charge to the sensing area, while the telomere's elongation brings negative charge, and we can sense these changes by measuring the sensor current. Here, the full lab-on-chip architecture is shown. The chip has an array of 1024 CMFET sensors, the readout and the analog to digital conversion circuitry, the potentiostat, the temperature control system, and a digital logic module which manages everything on the chip. Finally, the chip can communicate with the external environment through a serial interface. This image shows the complete layout of the chip. The sensors are organized in channels of 32 sensors each, and multiple readout circuits are repeated in each channel, and this allows for the execution of parallel measurements. The channels are laid out to allow a microfluidic system on top of the chip to deliver liquids and reagents on the sensors. The second part of the activities focused on the development of an integrated circuit for bacterial biosensing. Bacteria can have some advantages over more traditional bioreceptors like enzymes because they are easy to grow and can be genetically modified to be selective to specific chemicals. In this specific case, the chemotaxis process has been exploited to use bacteria as chemical sensors. When they explore the environment, bacteria like E. coli perform a random walk, changing their direction periodically. However, when they sense a gradient of an attractant in the solution they live in, they can change their movement by performing longer straight runs to follow this chemical gradient. This process is called chemotaxis, and it's used by bacteria to find nutrients or go away from toxic substances. Chemotaxis is a fast process that happens on a scale of hundreds of milliseconds. Moreover, chemotaxis is also very sensitive. In fact, bacteria can sense a change of just a few molecules of a specific attractant in their environment. These two facts make bacteria almost like an ideal biosensor. Bacteria can change the type of movement from straight runs to random walks by changing the direction of rotation of their flagella, which they use to move. If we attach a bacteria to a substrate by one of their flagella, we can detect its rotation because the whole cellular body will rotate. For this reason, an impedance sensor has been developed in which bacteria will be immobilized between a pair of electrodes, and the rotation can be detected through changes in the impedance between these electrodes. To bypass the effect of the double layer capacitance between the electrodes, high frequency measurements are required. To measure this impedance variation, the electrodes are stimulated by sine waves at a maximum frequency of 10 MHz, and the current that is generated between these electrodes is proportional to the impedance variation. This current is then converted into a voltage with a transimpedance amplifier. Then this signal goes into a mixer that can extract the real and imaginary part of the voltage, which is proportional to the impedance. Finally, the signal is filtered with a low-pass filter. So in the end, the output will be a low-frequency signal, which is proportional to the impedance variation. The picture on the left shows the chip layout, while on the right there is a photograph of the fabricated chip. We can see, for example, in the red box, the electrodes where the bacteria will be attached. This chip is currently under test. So, to conclude, we saw that the electronic engineering can be used together with other biosciences to give rise to biosensing applications. Many different applications exist, each one requiring different approaches and electronic design techniques. And finally, electronic platforms and lab-on-chip can be used as a cheap solution to replace complex and expensive lab equipment, and they can perform the entire measurement in a single device. Thank you for your attention.